this this reaction is likely to happen in this case. That wasn't the best explanation, but I think it's when it comes down to, especially OChem, I think in the MCAT, it's about having that understanding of stability and what that looks like in a mechanism. Yeah. Okay, glad I could help. Okay, so I just went over some of the ways in which we have nucleotide mutations, but mutations in the nucleotides can also cause an overall mutation in the DNA sequence. But they're also different types of mutations. So there are 4 main categories of mutations to understand and understand what impact they have. And it's not only important to know these mutations by name, but also understand how they impact the mRNA, the protein, and the organism's phenotype. So in our silent mutation, which is, so we can see over here, this is our wild type. This is what it is before any mutation. And these 2 strands over here are our DNA, and we're going to code, that's going to undergo transcription and then translation. And you're going to end up with these amino acids at the end of that. So in a silent mutation, which is our first one here, in a silent mutation, there's no change in the primary sequence. So we see this change over here, this point mutation here, where it was originally a TA base pair, but it changes into a CG. And generally that, even though we have this change, even though we have this mutation in the DNA, we don't actually see any change in the amino acids. They're both cysteine, nothing changes. So you often see silent mutations because the mutation is in the 3rd nucleotide of the codon. So that's why I went over sort of codons and that sort of stuff before we got into this part, because I wanted to emphasize that silent mutations often happen in that 3rd nucleotide of the codon. And that's a concept that's going to come back. So again, silent mutation, there's no change. In the primary sequence. For missense mutations, we see that, again, an amino acid, there's a mutation, but this time around, there's an amino acid that is changed, but it's only one. We don't see a change. All the other ones are the same. It's just one mutation that's changed. So there's only an impact on one amino acid. I hope the AA makes sense to folks, or that's easy to note. Okay, and then there's a nonsense mutation. So that's a premature stop codon. So the stop codon occurs instead of an amino acid. So remember I mentioned the stop codons earlier. So this is part of why it's important, because it's good to recognize when you have a stop codon. So it's not, it wouldn't actually stop over here normally, but the mutation causes the premature stop. And then lastly, there's a frame shift mutation. So the entire reading frame is shifted in the frame shift mutation. So so basically, because of that, usually you see almost every single amino acid change. So over here, what happened is that there was a deletion of one base pair, but because of that, instead of starting over here, or technically I would be looking at the wild type, instead of starting over here, you're shifted by one. So you're looking at this, and then so it's not only that first amino acid acid that's impacted. It's each and every single amino acid in each and every single codon after that. So you have the next three and then the next three. They're all going to be changed because of that one deletion. So frameshift mutations are often caused by an insertion or a deletion. So please let me know if that doesn't make sense. Okay, and someone messaged me about emailing them the PDF. I think I'm going to see, yeah, we have a practice question coming up, and I'll see if we have time for a break. So if I get a chance, I'll do my best to email it to you then. Okay, someone said there was an addition. There was an addition in which one? Was it the frameshift one? From ACT to ACC. Frameshift mutation. Sorry, I'll have to point it out to me. Top strand. Are you talking about the wild type one? So in the frameshift, you're saying that there was another mutation where it goes from ACT. Oh, and the top strand over here. Okay. ACT to ACC. Oh my gosh. Wow. What a good eye. I've been over these slides a few times and I never noticed that. Wow. Yeah. I think that might have been a mistake, but yeah, I don't think that was supposed to be there. But oh my gosh. Wow. That's a good catch. Yeah. I don't think there was supposed to be a mutation there. Okay, someone asked, is the stop codon based on the coding strand, not the template strand? Good question. We're going to get more into it as I talk about transcription, translation, but basically it's, actually I'm going to ask you to hold on to your question because I think it would not be a good use of our time for me to try and explain it here, but without like the visual aids and stuff. But when we get there, if you still have that question, please let me know and I can help sort things out. So for frameshift, someone asked about frameshift mutations and if it's just one insertion or one deletion. It doesn't have to be just one insertion or one deletion. It could be insertion or deletion of multiple base pairs. But I think like one insertion, one deletion is the most common way that you see that frameshift mutation happening, but there are other ways also for that frame to be shifted. Okay, so I have it all written out more nicely in this next slide. Okay, so I have a question for us. What is or what are the most deleterious DNA sequence mutation or mutations and why? So feel free to think about it, drop your answer in the chat. Okay, someone said frameshift because the entire rest of the strand has changed. Okay, okay. Someone else said frameshift, someone else said nonsense, it prematurely stops. Okay, someone else said nonsense as well. All right, yeah, exactly. Both of you, I think, I would say both of you are correct because yeah, nonsense will stop the complete production of a protein. So you might not have like most of the protein even created if there's a premature stop codon. And for the frameshift mutation, that'll change all the amino acids in the rest of the sequence. So you'll end up with a completely different protein. So I guess a follow-up question to that is what factors would you consider if you really had to decide? If you were to be given more information, what factors would you want to think about? So I'll let you think about that for a second. Okay, since that how much it affects the strand. Yeah. So you definitely want to factor in, and I can't think of it quite an example question for this, but one thing that I would recommend that you think about is the distance of the mutation from the end. So let's say there's a nonsense mutation. Let's say there's like a nucleotide sequence that's like 100 nucleotides long. Oh my gosh, I can't say words. 100 nucleotides long. If there's a nonsense mutation that happens nucleotide number 5, that means that's it. You have 5 nucleotides. That's your entire. That's it. You don't have anything else because it stopped it. It stopped further further translation actually. But you've lost everything after 5. Whereas if you have that same nonsense mutation, like nucleotide 97, you've lost 1 amino acid. So if you were to compare like a frameshift mutation, then that happened much earlier on, as compared to a nonsense mutation that happens much later on, those might be things to think about. You won't be asked to like compare exactly that, but it's just about like having the understanding of what factors impact create like this impact on mutations and create a larger impact, things like that. So I think that's just sort of a general tip I would have for bio, is trying to understand the relationships between everything. Even if like it's not apparent, trying to think about what potential impacts can something have. All right, any questions on that? Okay, so next we have a practice question. Actually, yeah, during this practice question, I can probably email this to that one person. Please email me if you still, or sorry, please send me a message if you still need it. And I will try to take a little bit of time and send that to you. But yeah, we have a practice question. I'll just give everyone a few minutes, a couple minutes to work on that while I send this email. All right, thank you everyone for your patience. I'm seeing a lot of B's in chat. Does anyone want to let me know, why did you think B? Okay, someone said because it just changes one amino acid and doesn't change the length. Same length, differing in one amino acid. Yeah, exactly. Yeah, just sorry. Yeah, one nucleotide. Um, all right. Did anyone say anything else? It's okay. I want to make sure that I'm explaining all the other options, especially if someone picked a different one. Okay, so I'll just go through it quickly. So nonsense and frameshift mutations can include premature stop codons, so that's definitely going to affect the length of the peptide chain. Insertions and deletions will cause frameshift mutations, so that sort of eliminates insertion, deletion, frameshift mutation, nonsense mutation. That's pretty much what we have. But yeah, that missense mutation is going to only change one amino acid. It's only going to swap out one amino acid, and it's not going to change the length of the peptide chain. If it were to like change into a stop codon, that would not be a missense mutation anymore. That would be a nonsense mutation. All right. Okay, so now I'm going to jump into transcription translation. If anyone has any questions from this last section, this would be the best time to ask those. Okay, I'm going to jump into it so we have time to go through more practice problems, hopefully. Okay, so one thing I wanted to really highlight throughout this was the difference between bacterial transcription and eukaryotic transcription. So in bacteria, there are multiple genes that are under the control of a single promoter. So promoters are basically these regions of DNA that can turn a gene on or off. And you're going to understand a little bit more about promoters as we go through transcription. Compared to eukaryotes, where there's only one promoter for one gene. So you don't have multiple genes controlled by one promoter. So it's a little bit simpler in that way. It's a little more complicated. We're going to see. So transcription occurs for bacteria in the cytoplasm. So this is something that I had mentioned earlier too, but yeah, transcription occurs in the cytoplasm because there's no nucleus for the bacteria. Whereas for eukaryotes, there's the transcription that occurs in the nucleus specifically, because that's where all the genetic material is. So RNA transcripts in bacteria are not modified after transcription. So you have your DNA, oh, missing a letter there, DNA to RNA, and that's it. You're good. You're good to go for your bacteria. You don't have anything else that you need to do. But for eukaryotes, you have your DNA, and you actually end up with your it's called pre-mRNA. And it has to go through post-transcriptional modifications prior to exiting the nucleus. And this is sort of what we had talked about earlier, where it has that poly A tail and that GPP 5' cap. And I'm going to talk a little bit more about that again as we go through this. Oh, I think that's the same slide twice. Okay. So in our bacteria, in both our bacteria and our eukaryotic cells, we have a promoter. And just after the promoter, we have this transcription start site. So we have the transcription start site just after the promoter for both the bacteria and the eukaryote. So what is the purpose of the promoter? I talked about the promoter. What does it actually do? So RNA polymerase, which is our enzyme that's actually going to do the transcription. It's going to be responsible for making the transcription happen. It's going to be binding to that promoter region for both the bacteria and the eukaryote. One thing I actually wanted to mention before I get too much into this is sort of a little bit more terminology. So let's say you have that RNA polymerase, which is going to be doing transcription. Just to know like relative terms, terms that tell us what is relative to something else. So something that's before the RNA polymerase or before like the region that you're talking about, this direction is called being upstream. And anything that's going down the other direction is said to be going downstream. That's for both eukaryotes, bacteria, but it's just some terminology that you're probably going to hear a lot of about where different elements in DNA are in relation to each other. So coming back to promoters and comparing bacterial promoters and eukaryotic promoters. So bacterial promoters have two essential DNA sequences. They have it's called like a -35 element and a -10 element. And the numbers basically indicate the number of nucleotides before the transcription start site, upstream of the transcription start site, if you will, upstream of the transcription start site, that these two elements exist. So this is 35 nucleotides before the transcription start site. This is 10 nucleotides before that transcription start site. And both elements contain a high number of AT base pairs, both the 35 and the 10. They both have a lot of AT base pairs. And the reason for that is that since AT base pairs have two hydrogen bonds, this is where it's sort of important to know that background knowledge, to have the deeper understanding of it. Since they have two hydrogen bonds between them, as opposed to 3 between GC, GC has 3. It's easier to pull apart the two DNA strands at this location. So you have lots of AT. I apologize for all of the scribbles, but hoping this is making it easier rather than harder. But you have a lot of AT nucleotides. And those are going to be easier to pull apart than having ATGC, or like a lot of GC base pairs. And then it's actually quite similar for the eukaryotic promoter, but they have a different sequence. They have a sequence called the Tata box. And that's because you have that TA base pairing. And again, the reasoning behind that is the same as with bacterial promoters, that AT base pair, it's going to have two hydrogen bonds. That's much easier to pull apart than GC. Okay, so I'm going to get more into transcription now. So transcription has 3 main steps. The first step is initiation. So in bacteria, there's a sigma factor. Should I change the color? I think I'm going to change the color. It's a little hard to see the promoter. Yeah. Oh. Okay, let's go orange. Yeah, I think that's good. Okay. So in bacteria, you have a sigma factor, which is a protein that helps the RNA polymerase find and get to that promoter site. So sigma factors are specific to the gene, but that's not something you need to worry too much about, but it's just something I wanted to mention. Okay, I'm debating whether to ask everyone this challenge question now or a little later. Okay, and then compared to that, eukaryotes have these transcription factors. So sigma factor is also technically a general transcription factors, but it's just a more specific term. But eukaryotes also have transcription factors, and transcription factors basically regulate transcription. And they can bind to the promoter region and recruit RNA polymerase. So that's exactly what sigma factor is doing. It's binding to the promoter and it's recruiting RNA polymerase. It's calling RNA polymerase to come and transcribe this area. this region, this gene. And yeah, sigma factor is a type of general transcription factor for bacteria and basically supports that transcription. But in eukaryotes, it's a little bit different because RNA polymerase actually needs general transcription factors to be able to bind the promoter. So sigma factor in some way is optional for the bacteria, but you absolutely need transcription factors in the eukaryote to be for the RNA polymerase to come and bind to the promoter. And it's just general transcription factors that it needs. So I'll be talking a little bit more about transcription factors a little later, but there are many non-general transcription factors that can activate or deactivate specific sets of genes, but those aren't always required for the promoter to be able, for the RNA polymerase to come and bind to the promoter. And lastly, there are many different types of RNA, but the main ones to know about are rRNA, which is ribosomal mRNA, mRNA, which is messenger RNA, and tRNA, which is transfer RNA. And we're going to get a lot more familiar with those too as we go through this. Okay, so my question before I go to the next slide, it's this one. So if we know that levels of sigma factor can be regulated through transcription and translation. So given that information, what might overall high levels of sigma factor indicate? What can it tell us? Okay, increased transcription of a specific gene. Yeah. And specifically, maybe thinking about the state of the organism. So sigma factor is seen in a lot of different genes. So it's, even though they're different sigma factors, it's like an overall term for a lot of general transcription factors in bacteria. Any other thoughts on that? Okay, someone said upregulation of a protein. So I didn't quite give you enough information here, but this is like a type of question or a type of idea that you would maybe practice with. Exactly. Sorry about that. Someone said bacterial growth. Exactly. So oftentimes sigma factor expression or activity is dependent on specific growth phase transitions of the organism. So I didn't tell you that, and you might not know that in a question, but if you're asked generally to sort of make a guess, that's a guess that you might want to make. That since it's a general transcription factor, it can impact the transcription of multiple different genes. It might, when we need a lot of transcription, we need a lot of transcription when you have growth in the bacteria. So if transcription of genes is involved in the growth is necessary, the sigma factors will be translated to allow for transcription initiation to occur. However, if transcription of genes is not required, sigma factors won't be active. Does that make sense? Is anyone lost on that? Okay, I know that can be a tricky concept, so yeah, just let me know if anyone's still confused on that or wants me to re-explain that in a different way. Okay, so moving on. So bacteria also contain a region called the operator. So this is unique to bacteria. So the operator is located between the promoter. It's located between the promoter and the transcription start site. repressors are a type of transcriptional factor that can bind to the operator and suppress the gene transcription. So repressors can physically block the RNA polymerase from transcribing genes. So if you have, let's say this is a repressor here, it would not overlap with the transcription start side, but oh yeah, let's say you have this repressor here. It can physically stop the RNA polymerase from continuing onwards and from transcribing the genes that are after that operator region. Activators, on the other hand, are transcriptional factors that can increase the transcription of a gene. And activators generally do this by supporting RNA polymerase to bind to the promoter region. So you'll see activators generally bind to that promoter region and helping with recruiting RNA polymerase. Inducers can activate or suppress transcription by interacting with a repressor or an activator. And repressors, activators, and inducers are all transcription factors that you might see in both bacterial transcription and eukaryotic transcription. But bacterial transcription has the unique operator region. So switching over to the eukaryotic side of things, eukaryotes have this mediator protein complex. So that's basically a multi-protein complex that functions as a transcriptional co-activator in all eukaryotes. So I know that's a lot of word vomit, but it's basically a multiple proteins put together that support transcription and activate transcription in eukaryotes. And the main function of mediator complexes is to transmit signals from the transcription factors to the polymerase. So these are our transcription factor binding sites. And we have these proteins here that are interacting with this protein in this protein complex and encouraging or discouraging transcription, because you could have activators or repressors bind here. So that's the interesting thing actually about eukaryotes, is that transcription factor binding sites in eukaryotes can be found near to and very far away from the promoter. So because of that, activating or repressing transcription factors can bind to these sites and then bind sort of to this protein complex and impact transcription. Whereas, again, comparing back to the bacteria, you have to have your activators and repressors all bind in this very small area, so the promoter region or the operator region. Whereas with eukaryotic transcription, you have a little more flexibility with the transcription, which also means more variation. Any questions on that? So I'm going to pause just for a moment to get a drink of water. Okay, someone asked, is it only located upstream? That's a good question. I don't want to give you the wrong answer, so I'm not sure. I want to say yes, but that's not a very confident yes. That's a good question though. If you want though, I can look into it and yeah, try and remember that. I'm gonna make a note of that. Okay, someone asked me to explain the mediator protein complex again. And then someone else also has a question about the mediator protein complex. Okay. In simple terms, or as simple as I can get it, the mediator protein complex is a group of multiple proteins sort of interacting with each other and either activating or deactivating transcription in the eukaryote. So because that complex is binding to the transcription the transcription factors and the transcription factor binding sites. Because of that, it's able to sort of encourage or discourage the RNA polymerase from transcribing. Okay, so I hope that explains it. Happy to re-explain that. So I said that the mediator protein complex will activate signals from transcription factors to, transcription factors to that, to the RNA polymerase, and ultimately, like, it will impact transcription. Yeah, it's known as a co-activator. Okay, someone said the eukaryotic picture shows 5' to 5' and 3' to 3'. Oh no, does it? You're right. That is a mistake. That's not supposed to be there. Thank you. These are not my images, but should have still cut this, but that's all right. Yeah, so this would be like 5' and this would be 3'. Thank you for catching that. It's okay. All right. Hope that made sense with the mediator protein complex. Yeah, it has the word complex in it. It looks complex, sounds complex, but it's for at least like how much you need to know for the MCAT. It's not too bad. Okay, I'm going to keep going, but please feel free to pause me, stop me if you have any more questions. So if you remember what I said last lecture, I talked about like a way to memorize the way in for DNA replication, how to remember like which direction DNA polymerase reads versus writes the DNA sequence. So that rule still, or that sort of memorization technique still applies here. So RNA polymerase, this is our RNA polymerase. Yeah, someone still remembers in the chat. But yeah, RNA polymerase is going to read from 3' to 5', 3' to 5', and it's going to write. So it's reading this template strand from 3' to 5', and it's going to write 5' to 3'. And the sort of memorization technique for that is that you learn to read before you learn how to write. You learn to read when you're 3. So that one comes first, 3 to 5. And you learn to write when you're 5 to 3. Yeah. And then someone had also asked last or 2 weeks ago about sense and antisense strands. So you hear that terminology specifically in relation to transcription. So you have, so there's the 2 strands and they have multiple different ways that they can be called. So there's the coding strand, and that's not the strand that's that the mRNA is complementary to. So there's the coding strand, it's also known as the sense strand, or can be just represented by a plus sign. And then there's the template strand, and that's what mRNA is complementary to. The template strand, also known as antisense strand, or represented by a negative sign. So I sort of answered this already, but something really important to think about here. What is RNA similar to? I'm saying similar, not the same, because there's a little bit of a difference, but which of the 2 strands is RNA similar to? Feel free to drop your answer in the chat. Someone said template, someone said coding. So it's actually, the wording was not the easiest there, but I didn't want to give away the answer. So it's actually similar to the coding strand. And I say similar, not the same, because our mRNA is going to have uracil instead of thymine. Okay, so why is that? Why is it similar to the coding strand and not the template strand? So if we remember, it's going to be complementary to the template strand. So if we were to give an example to this, let's say our coding strand is it's A G C A T C. This is 5 prime, 3 prime. This is our coding strand. Can someone tell me what our template strand is going to look like? Wait, G, think one more time. So like going from here to here, what would this one be first? And then go. So, oh, yeah, you would do it backwards, but I have this. Okay, sorry. So you gave it backwards. That's good. It's good that you're catching that. Yeah. So I wrote 3 prime to 5 prime, but you wrote your answer 5 prime to 3 prime, I guess. Yes. Okay. No worries. Okay. Someone wrote the response. Someone wrote a response including you. So one thing to think about is the template strand. This is still DNA. Template strand is still DNA. We're not at mRNA quite yet. So the template strand is going to be complementary to the coding strand, and it's going to include thymine. We're not at uracil. No worries, no worries. I just wanted to make sure that was clear. So from 3 prime to 5 prime, it'll be TCG, TAG. And yeah, Sophie, I think you gotten that right. It was just, I was misunderstanding the way that you had written it. Okay. And then what would the mRNA be? And I'm going to write that 5 prime to 3 prime. Anyone have any idea what the mRNA is going to look like? Okay, I'm saying, yeah, exactly. AG, CA, UC, exactly. It's not lined up nicely. Okay. So basically, it's if you compare the coding strand to the mRNA, it's very similar. They're very similar. They're almost the same. The only difference is that thymine and uracil. So that's why when I was asking, what is the mRNA similar to? It's most similar to the coding strand compared to the template strand. It's complementary to the template strand or the antisense strand. It's similar to the coding strand because it's almost the same. The only difference is that it's not a thymine, it's a uracil. Okay, hopefully that makes sense to everyone. Again, let me know if you want me to re-explain any of that. All right, and there was that example written out. Did I write thymine instead of uracil? I would not be surprised. So in the mRNA, I wrote, oh, at the bottom here. Yes, I did, didn't I? I thought I had some sort of reasoning behind that. I think maybe I was saying that the coding strand has thymine instead of uracil, but if you look at it, coding technically comes first, so it's uracil in mRNA instead of thymine. But yeah, I think that made sense while I was writing it, but not afterwards. I'll just pass that out. Okay, yeah. So next slide is just that written out. Okay. Also, I wanted to give everyone a heads up. We might not be able, I might not be able to get through all my slides in time. So I have a few practice questions, but let me know if you'd prefer to do practice questions or if you'd prefer for me to get through the translation content. I think that's pretty important. Material. Okay, some sub material. Okay, so lastly, you still have termination for transcription. So we've been doing transcription, it's been transcribing, we have this beautiful mRNA, but now we need to end it. We're done. We don't need any more. This gene, this genetic code it's been transcribed. So there are 2 different ways to terminate. Oh, actually, I think I'm missing a slide here. I was supposed to have something for eukaryotes. Okay, I'm sorry about that. Okay, so at least for bacteria, there are two different ways to end the transcription. So there's Rho-dependent termination. So there's this protein called Rho, RHO, which binds to the this Rho binding site. So this is the Rho binding site, and this is our protein Rho factor. And Rho factor then starts moving from the 5' to 3' direction. And it starts trying to catch up with that RNA polymerase. And the RNA polymerase might even pause to allow Rho to catch up. And basically what happens is that when Rho catches up with the RNA polymerase, it hydrolyzes ATP, and it uses that energy to pull apart the mRNA and the DNA, and basically releasing the mRNA and ending transcription. So you might even have the RNA polymerase might actually transcribe a region that codes for the Rho binding site. So once that Rho binding site appears on the mRNA, that's how the Rho factor is able to sort of latch onto that and then start that termination process. So if you think about it, it's actually pretty smart. It's like, hey, I know I want to stop transcription a few nucleotides down. I'm going to code for something that's going to summon this Rho factor to bind and stop transcription when I want it to stop. So it's pretty interesting in that way. Okay, and then the second way that you can terminate transcription in bacteria is Rho-independent termination. And that's also known as a GC hairpin loop. So C and G nucleotides, one thing to think about those is that, or actually, let me back up a little, yeah. So at the end of a gene, at the end of your DNA, there's a region that's very rich, and this is for bacteria again, there's a region that's rich with cytosine and guanine nucleotides. And there are a lot of, and since it's just cytosine and just guanine, the complementary CG nucleotides can bind together within the mRNA itself and create, and the mRNA basically folds back on itself and creates this hairpin structure that stops the RNA polymerase from continuing transcription. So because it creates this sort of like this rigid structure that forces the RNA polymerase to stop transcribing, and it pulls the mRNA apart from the DNA. Someone asked, does Rho factor enter the DNA polymerase, or does it just interact with the polymerase unlike mRNA? So just, sorry, a small correction. It's the RNA polymerase. So it's the RNA polymerase that's transcribing. I can't say I know. Actually, I do know. So there are a few different ways that this is, no worries. And there are a few different ways that this is described. But a lot of times it's described like the Rho factor is sort of like yanking at the mRNA and pulling it away from the DNA. And I'm not sure if that's like, if it gets inside of the mRNA polymerase or if it just like gets to the mRNA polymerase, but it's somewhere in that vicinity. So I don't have an exact answer for you there. And I know this is also somewhat new, so there's still like research being done on this and things like that. Yeah. Yeah. Yeah, it's beyond the scope of the MCAT, and if you get something on it, it'll likely be like a passage. I did have eukaryotic termination in here. I don't know. I think somehow it got deleted. That slide got deleted. And I'm sorry, but I can't tell you straight off the top of my head what eukaryotic transcription termination looks like. So I'm so sorry, I don't have that information for you, unless we want to take a couple minutes, but I don't think we have that time if we want to keep going. So that information is in the book, or if it's not in the book, you can definitely find it online. But if you want me to send you that deleted slide once I recover it, yeah, I can send it. Unfortunately, I don't have everyone's email, so if you want it, just drop your email in the chat, and I'll make sure to capture it and forward that on later. Okay, so I did have a practice question here, but I'm going to skip past it for the time being. Yeah, I'm going to skip past it for the time being, just so that I can keep going and see how much I can get through with translation. Yeah, and then if we have time or folks want to stick around for a while afterwards, then we can go through it. Okay, so now that we have our mRNA, what happens? Okay. Someone asked if I can tell them the answer. I think that's only fair. Oh, sorry about that. So I will tell you the letter. The answer is D for the question, and the question is in the slides. Okay, so if you remember, I'd mentioned earlier that eukaryotes are a little special what they have to go through. Sorry, it's just reading chat. Eukaryotes are a little special where they have to go through mRNA processing after the mRNA is transcribed. So you actually have pre-mRNA, which is the pure transcribed version. Nothing has been done to it. So it's not mRNA quite yet. And so I previously mentioned this when I went over the central dogma, but basically a 5' GPPP cap is added to the mRNA, as well as a 3' poly A tail. And there are these enzymes known as lysosomes, which cut before and after this intron region. So you have the exon, you have the intron. The exon is the region of genetic material that is valuable, that needs to be kept, and that is very important to coding. So you might get asked questions about, like, if we cut out this exon versus we cut out an intron, what is the impact on the genetic material, on the phenotype? So if you cut out an entire exon, that's going to be more deleterious than cutting out an intron, because introns, it's sort of in between genetic material that doesn't necessarily it's not translated in the end. So yes, lysosomes are made up of snRNA. So I mentioned that there are a lot of different types of RNA, so that's one type, plus some proteins, and it's a very witty name, SNRNPs, or I like to think of them as SNRPs. But yeah, basically they cut out this intron, so you're only left with the exons in the mRNA. And the intron that is removed, or the introns, actually, I should say, that are removed, are degraded, so that the nucleotides that are within that intron can be recycled. Yeah, someone said SNRP sounds like SNP. Exactly. That's why I thought it was a little witty. Whoever named that. Okay. So then you end up with this fully mature mRNA, which contains only exons, no introns, has a 5' cap, a 3 poly A tail, and it also usually contains this 5' and 3' untranslated region. So it's basically on both sides that flank the coding region. So I'm not quite sure what its purpose is. But if I were to guess, I would say that it's sort of that additional protective material to protect the actual coding region of the mRNA, because you don't want mutations there. You don't want things to be cut in that coding region, because that's where translation is happening. And if something gets messed up there, that's going to impact the protein that's translated in the end. And so with the different exons and introns, you actually also have alternative splicing in eukaryotes. So that alternative splicing happens when during the splicing portion with the spliceosomes, you have the option to pick and choose, like, and sort of mix and match which exons you can have in your mRNA. So you can have like all 3 exons. You can have exons 1 and exon 3. You can have exons 1 and exon 2. So you have a lot of different options there. And that also creates, because of that, that creates multiple polypeptides from that single pre-mRNA. This is one pre-mRNA, but you have a lot of different options, and that creates a lot of different options for different proteins. So sort of maximizing that protein or that genetic material and the capacity of that. So this is, again, sort of a summary slide for eukaryotes, where you have transcription, you add the 3' cap and poly A tail, sorry, 5' and 3' cap. Then you undergo splicing, and then finally, you have your mature mRNA, which undergoes nuclear export and exits the nucleus, and then that's where it can undergo translation. All right, I would usually pause for questions, but I'm going to keep running through. I want to make sure that I can at least get through translation before the end, before it hits 11. So for translation, translation can happen. For translation, you need a ribosome, and in the bacteria specifically, it's called the Shine-Dalgarno sequence, and that sequence helps to properly align the small ribosomal units. So there are actually 2 ribosomal units, and the one on the bottom is the small ribosomal unit. And it helps align that with the mRNA and make sure that everything's placed properly. And again, that's unique to bacteria. And there are initiation factors. I think this is in both eukaryotes and in bacteria. There are initiation factors that help stabilize and assemble the ribosomal units around the start codon. So like I said, there are two subunits for ribosomes, one large and one small. And the ribosomes used in prokaryotes and eukaryotes are different. So in prokaryotes, the subunit is 70S. In eukaryotes, both subunits together are 70S, and in eukaryotes, both subunits together are 80S. And the S is called a Svedberg unit. But basically, if you're essentially breaking it down and looking at what does it actually measure, it measures the surface area. So this is something that I would really recommend knowing. I've seen it come up on multiple practice exams, and I've definitely seen it on AMC material. So for prokaryotes, those 2 subunits are made up of, the large one is 50S, and the small one is 30S. So you might be thinking, 50 plus 30 doesn't make 70, that makes 80. So what's going on here? So like I said, Svedberg units measure surface area. So when you put that 50 and 30 subunit together, you're cutting out sort of the surface area that's in between, and you're decreasing the surface area. So that's why you end up with 70 instead of 80. So there's a difference of 10. And I'm saying that because I think that might help you memorize it. And then in eukaryotes, you have your 80S ribosome, and that's made up of 60S large subunit and 40S small subunit. So that added together is 100. So that's also over. And that's over by 20. So I would often have difficulty remembering the difference or remembering the numbers exactly. And yeah, I would often have difficulty remembering those numbers. So I try to remember that prokaryotes have a difference of 10 and that they have that they're overall they're 70. Eukaryotes have a difference of 20 and overall they have 80. So that would help me sort of back calculate and figure out what are the subunit sizes. Sorry, I need to pause for just a second to drink some water. All right, so just to reiterate, definitely would recommend memorizing those numbers and having like somewhat of an understanding of why those numbers exist. So in eukaryotes, the start codon AUG codes for methionine. And that was something I'd mentioned earlier as well. And in prokaryotes, the same codon codes for N-formyl methionine, which is F-met. So a slightly different version. But good thing is you just have one codon to memorize AUG. Okay, so for translation, there's the codon that we know about. We've talked about the codon, and then there's the anticodon. So actually going up, this blue structure is called the tRNA, transfer RNA. So that transfer RNA is holding an amino acid. It's holding the amino acid and it's coding, it has this anticodon that is typically exactly complementary to the codon. And that basically tells, okay, which transfer RNA is matching exactly with the codon, and that's how we're going to get the right amino acid to match up, and we're going to have the right amino acid in this peptide chain. However, it's not always exact that the anticodon is exactly complementary to the codon. So if we remember, we have this chart again. We look back at this chart, we remember that for the most part, changing the third nucleotide on most codons doesn't change the amino acid. For most of them, it doesn't change the amino acid. And this is useful for translation because it means that we don't need to be so careful about that last position on the codon. Because if we look at this, again, there's 64 combinations. That means you have to have 64 transfer RNAs ready to go with their amino acid attached to just come in and be able to add the peptide, add the amino acid to the peptide chain. 64 is a lot, and having fewer means that it's going to be in a more efficient process. So that third position is known as the wobble position because it doesn't always have to be exactly complementary. So for many amino acids, the third position can be ignored, or for many codons, the third position can be ignored because you might end up with you'll probably end up with the same amino acid. This is not always the case, but for many it is. And that reduces the need to have all 64 combinations of the tRNA and anticodons available. You can have fewer than 64, and because of that wobble position. So coming back to translation, we have this this transfer codon. Oh my gosh, I'm sorry. This transfer RNA in the P site, and this had detected that this is the start codon, and this is our methionine. So now we have new tRNAs enter the ribosome at this, what we call A site. And we can think of that as the landing position. that's where new tRNAs land. So this complementary tRNA, this one, you can see it doesn't exactly complement that C, those Cs. That would be typically C and G, but it results in the same amino acid, so it's fine. So the complementary tRNA enters that A site and binds to the matching codon. And this process does take energy and in the form of GTP. So that first amino acid, the M, the methionine, then moves on to the new tRNA and forms a peptide bond with the new amino acid, which is the C, yeah. And then the mRNA as a whole, so the mRNA, not the ribosome, but the mRNA shifts forward by a codon. So it moves itself so that the ribosome is exposed to more 3' mRNA. So basically, if you're looking at the mRNA alone, the mRNA itself moves this direction so that the ribosome stays around in the same location, but it's seeing more genetic material, more mRNA. And then the empty tRNA moves to the E site. And then that empty tRNA basically leaves the ribosome, and So basically this happens over and over and over again until you have your fully formed peptide chain, until you see the stop codon that says, okay, stop. But I'm going to talk about that in just a second. And so if you want to see the order, it goes like this. This is 1, 2. This is just continuing the same thing that we're talking about, just elongation. Yeah. And then that M that we have in the beginning, that's our that's at the N terminus of the polypeptide chain. And the T is at the C terminus, just to connect to other concepts. And then termination basically happens when that stop codon in the mRNA enters the A site specifically. And stop codons are recognized by proteins called release factors, which fit into that fit into the P site. And those release factors then cause a reaction that separates the chain from the RNA, from the ribosome. And then your new protein or your new peptide chain is released. Okay, I'm gonna keep going, just 4 more slides, and then I might try and see if I can wrap it up there. Okay, so you can have ribosomes in 2 forms in eukaryotes. You can have free ribosomes, which translate mRNA into cytosol. They exist in the cytosol, just in this space in the cell, and that's where they do their translation process. And then you also have membrane-bound ribosomes, and those generate peptides directly into the endoplasmic reticulum. So you might remember rough ER, or rough endoplasmic reticulum, and those are called rough because they have ribosomes attached to them. And those ribosomes are generating this polypeptide chain directly into that endoplasmic reticulum. And again, this is unique to eukaryotes. So once you have that polypeptide chain created, there's often a post-translational process, especially if that polypeptide chain was created directly into the endoplasmic reticulum. So the completed peptide then goes through post-translational modifications, such as help with protein folding and other changes to the structure. and this is done by the endoplasmic reticulum. And then the ER, or endoplasmic reticulum, often labels peptides with a destination, assigning them to go to locations like the nucleus, the Golgi apparatus, secretory vessels, other locations, other organelles. And the endoplasmic reticulum also performs quality checks on proteins, and it sends the incorrectly produced proteins to be degraded. The Golgi apparatus might then, if it sends it to the Golgi apparatus can package the protein to be secreted from the cell and to be secreted from the cell or embedded in the plasma membrane or something else somewhere else. On the other hand, peptides that are produced by free ribosomes that are just in the cytoplasm, those peptides can remain in the cytosol or they could be sent to the nucleus. And those proteins that are being sent to the nucleus will have a nuclear localization sequence attached. So it'll basically help it zero in on getting to the nucleus. Okay, so I did have more slides. That's all I have for translation transcription. I had more slides on epigenetics and more practice questions that I didn't get to, but I want to make sure that I'm answering any questions with our remaining time. So any questions about any of that? Quality check. Someone asked, what does quality check mean? It's basically, I was just trying to show that the endoplasmic reticulum can also check for the quality of the amino acid and check that everything's going all right, like the sorry, not the amino acid, the peptide chain, which is made by amino acids. So we can check that the peptide chain is made correctly, it has all the correct components. So that's what it means by quality check. Yeah, it's doing this like sort of checking process and then also sending it out, getting it so that it's ready to be used as a protein. Oh, that's a good question. Why don't mutations get a negative quality check? I can't say I'm very familiar with the quality check process. Yeah, I'm not sure how much it checks for mutations. I don't know if it checks for if the protein is, because it's not, if you think about it, it's not going to be like comparing to the mRNA. It doesn't have the mRNA to compare it to, or it doesn't have the genetic material there for it to compare it to. So I see quality checks as like being more that like it has been formed correctly, like the peptide bonds are where they're supposed to be, that the things like that. But that is a good question. I think mutations, there's more of like the DNA repair system. I don't know if I'm not familiar with if there are any other systems outside of that DNA repair system that can impact, that can check for mutations, but I would not be surprised if they are. All right, so I'm down to stick around for like half an hour or so, and spend this time however works best for folks. So if we want to go over more practice questions, if you want me to like go over the material that I missed earlier, if you want me to dig out that one slide that I had accidentally deleted, I can do that too. But yeah, if you have to go now, then that's completely fine. What is the best resource to learn bio since it's not TBR? Are you just asking for like how to like what's the best way to like go over this material? The book set. Okay. Okay, let me answer this question, then I'll go over that slide. So are you asking for like book set recommendations or? Just want to make sure I'm understanding the question. Kaplan versus exam crackers. So personally, I went mostly off of TBR material when I was like before taking full length. Then once I started taking full lengths, I if there was something that I didn't find in like in my notes that I took from lectures or in the textbook, I would just Google it. It's not always ideal because you don't always get the best, you don't always have like a comprehensive answer. But there's a lot of good online resources out there that explain things very simply and very straightforward. So for me, I think Khan Academy is a really good one, especially their articles. I think I actually use some of their diagrams. They have really good diagrams. Um, I have the Kaplan books. I didn't use them too much because honestly, I felt like they covered the overview, but for some of the details that I would get stuck on for full lengths or some of the very like nitty gritty stuff, I didn't feel like it had all the content, which it's not possible for a book to cover everything that the MCAT is going to have. Um, yeah, but I guess if your question is like an overall feel of the material, I think Kaplan um, is pretty decent in giving you like the important things that you have to understand. I think TBR definitely goes into too much detail sometimes, but it's also I think it's a good exercise to try to go through the book and piece things together and try to like go into the book with like a problem solving sort of mindset instead of like, okay, I'm just going to read this and learn it. Instead, like I'm going to read this and actively engage with this material and try to learn it by like trying to piece together the different content. Um, I can't say that I know of like a specific book set that like does a very good job at applying what does this look like in the MCAT or in like actual practice problems. And I think that's just something that you're gonna get used to and you're gonna figure out as you do practice problems and as you take like full lengths. Yeah, I hope that helps. Um, all right, if folks can give me like 4 minutes, I'm gonna go through and see if I can find that deleted slide. Okay. Thank you all so much for your patience. And again, if you have to go, no worries. And feel free to email me if you have any questions. And I have office hours on Monday. All right. Just going to take a couple minutes. All right, sorry about the wait. Okay, so I actually wasn't able to find the slide. I don't know what happened, where it went. But I do have like what I was going to write on the slide. I'll just paste it in the chat. But it's not very much. So again, this was eukaryotic transcription termination. So it was that this is a field where there's still like research being done, so they don't have a lot of information on it. But basically, termination begins when a polyadenylation signal, which is like a lot of A's, appears in the RNA transcript. And actually, if we go back, so this is transcription. Okay, it's not exactly the GC hairpin lip; it's different. But if you remember, the eukaryotic RNA ends with a lot of A's because it has that poly A 3' cap. And basically, that 3' that poly A 3' cap is also like how termination occurs. So they're not really familiar with exactly the mechanism of it, but basically it sends a signal once that's transcribed. Once that's transcribed, it sends a signal that's recognized by RNA polymerase to release the transcript from the DNA. So I'm sorry though. I feel like that was a lot of buildup to something that was very short. But for folks who were able to make it, I guess I'll just email them that very small blurb. But yeah, that was pretty much it for that one slide. All right, so I can stick around for another half hour. But yeah, I'm glad. I hope this was able. I hope I could help. I hope this was helpful. Please feel free to email me, come to my office hours if you have any questions. I'd be happy to go over passages. But yeah, if you have any questions or if you want to, my office hours are Monday 11 a.m. to 1 p.m. This Monday, and it might be a little. Yeah, I can go over epigenetics if folks want to do that. But no worries again if you have to go. Okay. Officers, here, let me type it in the chat. Oh, perfect. Thank you, Jonathan. Okay. So for epigenetics, this is just a very small part of sort of gene expression. But this is specific. This is generally more seen in eukaryotes. Yeah. So one of the important things that we learn about in epigenetics are histones. So there are these histone proteins that DNA is coiled around. And another way to talk about these histones is nucleosome. So nucleosome is basically this bundle of 8 histones. So each of these sort of gray blobs here, each of those is a histone. And 8 of those histones plus DNA make up a nucleosome. So nucleosomes are made up of DNA wrapped around histone proteins. And positively, so DNA, again, remembering what is DNA made up of? DNA has a negatively charged backbone. So DNA negatively charged. So one thing to think about is how does that interact with things around it? So histones often have positively charged amino acids, so arginine, lysine, histine. Positively charged amino acids and negatively charged DNA. So the positively charged lysine in particular attracts the negatively charged DNA, and that coils the DNA more tightly. So you can see sort of what that might look like, where you have these lysines with these different structures at the end. And so again, the positively charged lysine attracts DNA, attracts the negatively charged DNA. Actually, I think I'm going a little out of order here. So you can actually change the way in which lysine interacts with the DNA by choosing to acetylate the lysine or not. So lysine is located on those histone tails, and acetylating the lysine, this positively charged lysine, acetylating it makes it neutrally charged. Oh my gosh, I can't spell. Okay, it makes it neutral. And so now that this lysine is neutrally charged, there's not as much interaction between this lysine and DNA because it's no longer positively charged, and so it's not attracting the negatively charged DNA as strongly. So this basically decreases histone DNA interactions. So it promotes a looser overall structure, and that looser overall structure, it was in the slide before, that looser overall structure is known as euchromatin. And this, because it's looser, the RNA polymerase is able to access it more easily, which means that transcription is happening. The more transcription is happening, which means the more translation is happening. So you're creating more, that gene is being transcribed more. So again, acetylating those lysine, the lysine that's on the histone tails will increase euchromatin. It'll increase that looser structure, and it'll increase gene transcription and expression. So that's a really important relationship to understand. So there's acetylation, where it will promote euchromatin. But what if you do the opposite? Let's say you have a lot of acetylated lysine, and then you deacetylate. If you deacetylate, then you're going to end up with, it's going to be positive again. And that's going to increase heterochromatin, which is the type. Heterochromatin is the tightly packed and tightly wound DNA around the histones. Okay, someone asked, I don't understand how it increases transcription. Okay. So let's forget about acetylation, deacetylation for a second. Let's just think about DNA being tightly wrapped around these proteins. So it's very, very tightly coiled. So if I were to draw this out, it would be like this. Lots of tiny coils, like very, very small. Versus, so this is our heterochromatin. Very tightly packed. Versus euchromatin, so loose. I'm just going at the extremes here to make this more understandable. It's very loosely packed. So that means that your RNA polymerase, this is not to scale, your RNA polymerase, when it comes in and it tries transcribing these regions, it's not going to be able to access the DNA in the heterochromatin DNA as much as it would the euchromatin DNA because it's so tightly packed. Some of it is like wound inside, some of it is it's just so inaccessible. So it means that transcription is not happening and there's not as much gene expression. Versus if you acetylate that, you acetylate it, you make it so that the DNA is it's loose. It's not there's a lot of room between the histone and the DNA. It makes it so that it's possible for the RNA polymerase to go in and transcribe these regions. And it actually increases transcription and it increases expression of those gene regions. So this is something that the reason that I'm pointing this out so much and putting so much emphasis on this is because I personally had this come up on multiple like practice questions. And I remember I always struggled with this, always forgetting like, which way does it go? Does acetylation do this? Does acetylation like, so I would always personally, like when I was studying, I would always try to just memorize like, okay, acetylation means more euchromatin. Acetylation means more euchromatin. Deacetylation means more heterochromatin. But I still would never remember, which is why I wanted to really explain like the mechanism behind it so that you can sort of understand it and memorize it through that. So remembering that acetylation means that your default lysine is being acetylated. It's being made to be neutral. And because of that, you're decreasing those histone DNA interactions, you're increasing transcription. And if you want a way to memorize, because I would always also get confused between heterochromatin and euchromatin, I would think of it like euchromatin is like you want to be relaxed. You want to be relaxed doing your MCAT. You want to be chill and you want to be loose. So because of that, euchromatin is loose and the opposite heterochromatin is tight. Someone asked if I could please repeat what HATs and HDs stand for. I actually didn't explain that, but HD is histone deacetylation and HAT is histone acetylation. And you don't need to know like exactly what it stands for, like what it is, but it's basically, it'll likely be like an enzyme that's doing this action. Why would deacetylation be needed? That's a good question. So actually, I can explain, no worries. I can explain why deacetylation and acetylation are needed. So looking at this in the broader picture, I want you to think about what is the purpose of having these options between heterochromatin, euchromatin? What is the ultimate impact and why is that important? No worries, it happens. Wow, look at you go, Sophie. I like that. Deacetylation leads to heterochromatin, which is tight. Nice. Yeah, but I want you to think about, I'm asking this because I want you to try and think about this a little more actively before I explain this. What is the purpose? What purpose does heterochromatin and euchromatin serve? Like, why is it even important to have those two different forms of DNA? And anyone can answer this, or at least give it a guess. So over here, we see that looser DNA upregulates transcription. Okay, someone said because some genes don't need to be translated as often as others. Yeah, exactly. So some genes you might want them to be. For example, like let's say you're undergoing growth. There's like a growth phase happening, and there are some genes that you need to produce like material for like DNA or cell membranes or DNA polymerase. So you'd want to increase transcription and translation and expression of that area. You want more of that. So how do you control the amount of translation or the amount of transcription of different areas of the DNA? You do that partly using heterochromatin and euchromatin, and by regulating heterochromatin and euchromatin, basically the status of the DNA. So if you want something to be upregulated, if you want something to be expressed more, you're going to want to promote the euchromatin version of the DNA. To balance the gene expression circumstantially, depending on what proteins may be needed. Yeah, exactly. Exactly. That's the purpose. That's like, yeah, I guess I didn't really explain how this fits into the context of epigenetics. So epigenetics is like how your DNA responds to the environment and responds in any given moment. So for example, you might often hear about like people get gray hair because they're stressed out. This is just an example. People get gray hair because they're stressed out. Maybe the pigment, the black pigmentation, this is not actually like the reason, but this is like an example. Maybe the black pigmentation, because of the stress, there's something that's causing histones to be deacetylated, and it's increasing heterochromatin, and it's down-regulating transcription. So there's less of that black pigment being produced, which is creating like the white pigmentless hair. That's just an example. Does that make sense? I want to make sure this is really, really clear because I think this is a really important concept to understand. What takes prominence in transcription? Downregulation, methylation, or decylation? Oh, you're ahead of me. Okay, I haven't talked about methylation yet. Methylation has this whole mechanism that I am honestly not too familiar with. I wish I would be able to, I could like explain the whole thing, but I don't quite understand it myself. But what you do need to know is that histone tail methylation, or actually this is actually a little bit just misrepresented a little bit, because the methylation actually happens to the nucleotides. This should be. The methylation actually happens to the nucleotides, not the histones, if I understand correctly. So histone methylation increases heterochromatin. So it tightly packs the DNA and it down-regulates transcription. So in some ways you can think of methylation as opposite to acetylation. So you said what takes prominence in transcription? Downregulation, methylation, or deacetylation? That's a good question. I would have to say it depends on a lot of different factors, because there are like transcription factors, and there are different factors that are impacting the methylation and deacetylation rates. And that might be different for different genes. It might be different for different things. So I don't have an I don't have an answer exactly for when methylation and acetylation is used. And I think that's a little out of the scope of the MCAT too. You won't be asked to like compare methylation versus deacetylation really. But it is important to have that concrete understanding of the role that methylation, acetylation, deacetylation, all of those play in DNA regulation and DNA transcription translation. I hope that somewhat answers the question, even though it didn't quite. Someone asked me to explain again what HAT and HG mean. You don't need to worry about that. Honestly, I should have taken that out. But basically HD is just representing like histone deacetylation. And HET is like representing histone, but it's going to be like an enzyme that's doing that. So it's actually, it probably actually stands for histone deacetylators and histone acetylator. And yeah, but yeah, that's not something that you need to like memorize. The really important takeaway from this is, again, that relationship between these different things. So let me see if I have any questions. I think I put questions in here to test you all. Okay, this should be pretty straightforward. But I guess it's just a way for you to test yourself that you understand these concepts. So does histone acetylation promote euchromatin formation or heterochromatin formation? Answer sort of on this page, but yeah, I said histone acetylation. No worries, no worries. Yeah, euchromatin, exactly. Yeah. Okay. I think most of my questions actually are pretty straightforward. I guess could someone, if I think for me, it would often help to explain things back to other people. So if anyone wants to do that, I'll leave that open. So I'll ask the question, and then if you want to explain it back to me, you can do that. Or if you don't, that's also fine. But um, what is the impact of histone deacetylation on DNA and gene transcription? And talking like thinking more about like not just what is the relationship, but why does this happen? Decetylation decreases transcription when you don't need something to be transcribed because it's tightly wound. Yeah, exactly. And why is it tightly wound? Yeah, because the positively charged lysine, so deacetylation, yeah, positive lysines are attracted to the negatively charged DNA. So deacetylation sort of releases the lysine and exposes its charged site, and it increases those histone DNA interactions, and it makes it so that there's more attraction between the lysine and the DNA. And as a result, DNA is wrapped more tightly around histones, and the heterochromatin, or the packed form of DNA, is increased. And because of that, that down regulates gene transcription and basically results in gene silencing. Yeah, but in terms of like content, I have like more practice questions, but that's all I have in terms of content. So I can stick around for like 10, 15 more minutes. If folks want to, if you have something in particular you want to do, but I'm pretty much done with the actual content. Yeah, and if you have questions about like bullying, practice exams, how to study, anything like that, that's all. Oh, thank you, Sophie. Uh, yeah, we can do questions. Yeah, but if you have like any questions on any of that as well, we can always, I'm always down to answer those. Okay, I don't think we Yeah, I think we had to skip over this one. We went over the first question. Okay, Marissa, feel free to ask your question. And the passage in question are also up, so if folks want to work on that, go ahead and do that as well.
Oh, can I just say it over? Okay. I was just wondering if there's anything particularly helpful that you did in regards to like learning how to break down bio passages. Because I feel like the TBR questions are a little more like specific detail-y. So it's like content that's like really specific details that are like really nitty-gritty. And I feel like the full length ones are more like, do you know how to like read this graph and like understand what's going on here? Is there anything that you found that was particularly helpful for you in regards to that?
Yeah, so sorry if we're talking over folks who are trying to answer the question, but yeah, that's a really good question, and that's something I noticed with TBR too, especially like the first time I was like exposed to the material. Actual MCAT is not as much like content focused, but like, can you apply this content? Can you interpret graphs? Can you like, do you understand this like weird experiment that we're doing? So for me, at least, I know a lot of times when I got stuck, it was like in understanding what is the passage even talking about. So I would have a few different strategies, and I would sort of change it up based on the type of passage and the type of question. So there were some passages definitely that I got that were just like, here's this background information about diabetes and insulin and this. I'm like, okay, I already know what diabetes is. I already know like this basic information. Maybe I'll skim it, but I already know what it is. So then I would go to the questions, and some of a lot of the times the questions were like, you needed the passage, or like the passage would help you, but you could do it independent of the passage as well.
Right.
Those are a little more rare. But a lot of times those were like, apply the knowledge that you have to this context.
Like the pseudo discrete ones, basically.
Yeah, pretty much.
Okay.
Yeah. So for those ones, it's like, okay, I'm not going to waste my time like trying to read the passage, but I learned to get a lot better at interpreting graphs and tables. So a lot of times I would like go through the content and like maybe not understand it, but then like, and maybe, so I guess if I were to break it down, I would say step one, this is just like sort of a blanket statement. I would say step one, identify what the variables are in the passage. Identify like, what are the things that we're talking about. Step two, identify the relationship between those variables. And this is something you might want to think about while you're reading it, so that you're not like reading it and it's not just going through your brain, because that would happen with me a lot, that I read it and I'm like, I have no clue what I just read. And I would go and read it. Yeah. And then I'm like, okay, I'm just wasting my time now. So taking that like active approach of like, okay, I need to identify what are the variables, what are the relationships between the variables. And a lot of times you would find out and you would understand the relationship through the graphs, or you would have to interpret the graphs to tell them what is the relationship, or interpret the graphs to identify the relationship and use that understanding of the relationship to answer the questions. So for those questions that are not quite discrete passages, and that are very experiment heavy, that are very table research, chart heavy, spending a good chunk of time, especially, I mean, if they have a lot of charts and tables, they might not all be relevant. They might give you extra information. So I wouldn't go through all of it, but maybe the first couple, just to understand, again, what are the variables, what are the relationships between the variables? And I often like write it out on my like scratch paper so that I have it in front of me and I'm not like, oh, is it this way or is it this way?
Okay, sounds good.
That's very
I'll
like
definitely
generic.
No, that's okay. I'll definitely try that and see how that goes, because we're having a bit of issues, but that's okay. There's still time. Thank you. I appreciate it though.
Of course, yeah. And yeah, I think just the more exposure you get to it, the more I know a lot of times like you see it and you're like, you freak out a little bit. You're like, I have no clue what you're saying. But once you start breaking it down in that way, then you get it, you see that passage and you're a little more calm going into it. So a lot of it is just like practice experience, getting more familiar with seeing like content that you have no clue. Because that's the point. Like you won't know everything. You're not supposed to know anything. They're trying to freak you out a little bit. It's a big part of it is a mind game.
Right. Okay. All right, cool. Thank you.
Of course. All right. Sorry, folks. I hope you were able to work on the question. If you have a response, feel free to drop in the chat. Otherwise, I can give a couple minutes to go through it for yourselves. Yeah, you got it. I don't know if there was anyone else who was working on the question, but yeah, the answer is D. Do you want to go through the logic of it, or are you feeling good with it? Or does anyone want me to explain? I think this one can be very easily be a tricky one, but I'm glad that you caught everything. Yeah, but basically, we have the coding strands that we're given, and we just translate that into the template strand. But a mistake that a lot of folks make is that they forget about the directionality of it. So once you find its complementary base pairs, you write those out, but that's, if you do it directly under, it's going to be 3' to 5', not 5' to 3'. And all of our option choices are all 5' to 3'. So then you would just flip it. So this is actually very similar to what had happened earlier when we were going over some of the questions. But yeah, you just flip it and then you end up with option D. And then there's this next slide in case you want to go through like all of the steps of what that would look like. But it seems like we've got it. All right, we can go to more practice questions if people want. I have time for, actually the next one is a full passage. Okay. Okay, let's just do those ones and then, yeah, we'll call it a day. Hopefully you have the slides in front of you, because I wasn't able to fit the passage and the questions onto the same slide. So it's slide 66, 61 for me. It might be like somewhere in the 50s for you.